In this last video for chapter 5, we're going to be looking at nucleic acids. Now, um, I'm going to keep this shorter than the other sections because we're going to come back to nucleic acids in very much detail when we're covering genes and transcription and translation. Um, Nucleic acids transmit and help express hereditary information. The amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. And genes are made of DNA. DNA is a nucleic acid made up of monomers called nucleotides. Now, what are the roles of nucleotides or nucleic acids? There are two types of nucleic acids. There's deoxyribonucleic acid, commonly called DNA, and then there's ribonucleic acid, commonly called RNA. DNA provides the directions for its own replication, and DNA also directs the synthesis and creation of RNA. And there are several different types of RNA, for example, messenger RNA, transfer RNA, ribosomal RNA. And so there are these different RNAs which are created from the directions from DNA, are used in the process called of gene expression, in that the creation of proteins. And so this is just a summary of the process or the uses of nucleic acids. So DNA is the information carrying molecule. Um, Basically, it's the leader, it's the one that tells everyone what to do. And then messenger RNA is created from DNA. That messenger RNA is then read, and proteins are then created from the reading of the messenger RNA. So we covered new proteins and how proteins are so important. Well, it is the DNA that is the carrier of all of this information. Each gene in a DNA molecule directs the synthesis of a RNA molecule. The messenger RNA will interact with the cell's protein synthesizing machinery, which we call ribosomes. And then the ribosomes will read the messenger RNA and then create protein. The flow of genetic information can be summarized as DNA creates RNA, which then creates protein. And it is that process that has led to the creation of all of the cellular material that exists. So what are the components of nucleic acid? Nucleic acids are polymers called polynucleotides, and each polynucleotide is made up of monomers called nucleotides. Each nucleotide has a nitrogenous base, a pentose sugar, and one or more phosphate groups. The portion of a nucleotide without a phosphate group is called a nucleoside, which is less common, but nucleosides are a nitrogenous base and sugar. Now, there are two families of nitrogenous bases, the pyrimidines or the purines. The pyrimidines have a single six carbon structure. So cytosine, thymine, and uracil are examples of pyrimidines. Purines, such as adenine and guanine, have a six-member ring fused to a five-ring member. And so I'm going to show you guys that in the image in a second. In DNA, the sugar that is used is called deoxyribose. In RNA, the sugar is ribose. And so here are the four common nucleotides that will form DNA. So you have cytosine and thymine as the pyrimidines, and then you have guanine and adenine as the purines here. So this is just showing you the chemical structure of a polynucleotide. So this is one side of a DNA molecule. And so each nucleotide is composed of a phosphate group, a pentose sugar, and a nitrogenous base. Now, depending on what it is, the nitrogenous bases can be different. So, for example, in the pyrimidines, you have cytosine, thymine, and uracil, where thymine is only found in DNA and uracil is only found in RNA. In the purines, you have the two ring structures, adenine and guanine. And then the sugars, the pentose sugar can, depending on what it is, if it's DNA, it's deoxyribose. If it's RNA, it's ribose, which they're very similar. They're all very similar, but they carry different information. So nucleotides are linked together by what they call a phosphodiester linkage to build a polynucleotide. So basically, it's a phosphodiester bond. Remember in proteins, the bond between amino acids was called a peptide bond. Well, the bond between nucleotides is a phosphodiester linkage. A phosphodiester linkage consists of a phosphate group that links the sugars of the two nucleotides. It is these linkages between these bonds here that creates the sugar phosphate backbone of a DNA molecule. And this is unique. The bases that will attach to this backbone is unique depending on the RNA that's being read. DNA molecules have two polynucleotides spiraling around an imaginary axis, forming what we call a double helix. So the structure of a DNA molecule is like taking a ladder and twirling or twisting that ladder to form a double helix. The backbones of the molecules will run in opposite directions. They run from what they call 5' to 3' directions. And this arrangement is called antiparallel. So one DNA molecule can include many genes. And so we're going to get to an image of that. I want to just show you. I guess I don't have a good image of that. How it goes from 5' to 3' here. And if we was to put the other side of the DNA molecule bound to it, it would run in the opposite direction. Only certain bases of DNA can pair up together and form hydrogen bonds. Adenine always binds with thymine, and guanine always binds to cytosine. So your A's and your T's will bind together, and your G's and your C's will bind together. And it's always like that. And so this is called complementary base pairing. Um, this feature of DNA structure makes it possible to generate two identical copies of each DNA molecule while only needing one half of the molecule. RNA is different from DNA in that it's not a double-stranded molecule, it's a single-stranded molecule. And instead of RNA having thymine, it has replaced thymine with uracil. So uracil is a different base, and it's thymine is not found in RNA. While DNA always exists as a double helix, RNA are more variable in form, and they're single strands, and they're not actually always helical. So this is showing you the differences between DNA and RNA molecules here. So in DNA molecules, remember, it's the ladder that's been twisted. It's the double helix. So imagine if you could take a ladder and twist it. And this does show you the 5' to 3' and the 3' to 5' sides of the molecule. And the backbone has the nucleotides. And this is showing you that C's and G's are bound together and A's and T's are bound together. And that's the only way you get those kinds of molecules. RNA is not in the helix. It's more free floating, it's linear. And so they are able to form a lot of different structures. It's not just one singular structure. Um, RNA needs to be more flexible because it needs to be read by ribosomal proteins that will create other proteins. And we'll cover a DNA in a whole lot more, a lot in depth when we get to um, the genetics chapters. The last part of this is about genomics and proteomics. Genomics and proteomics have transformed biological inquiry and application. Basically, genomics and proteomics is the study of the human genome and the human proteins that exist. Once the structure of the DNA and its relationship to amino acid sequences are understood, biologists are then able to decode genes by learning their base sequence. The first chemical techniques for DNA sequencing was developed in the 1970s and has been fully refined since then. We have fully genomed or determined the genetic structure of some organisms on the planet. It's enlightening to sequence the full um, DNA sequence of an organism. The rapid development of faster and less expensive methods of sequencing was a side effect of the Human Genome Project. So not only have we been able to identify the genome of humans, but we're starting to do that with a lot of different other species. So many genomes have been sequenced and generating large sets of data. It's important that we are able to learn the genome of some organisms. While it may seem like science fiction, but learning the genome of some organisms, if they so happen to go extinct, will have the information that is needed to actually build them again. So bioinformatics uses computer software and other computational tools to deal with the large data set that comes from learning the genomes of organisms. And so being able to learn the genes and learn how um, genes work, how they're turned on and off, learning about the different genes between different species is called genomics. And then learning about the large set of proteins that are able to be produced from these genomes is called proteomics. And so with this information, we're able to make connections and the contributions of learning whole genomes and learning about proteins of biology is that we're able to bridge gaps between paleontology. We're able to show evolution between species such as the hippopotamus and the short-finned pilot whale. We're able to make advances in medical science. For example, we're now able to offer gene therapy. We're able to offer gene counseling for couples to say if they want to try to have a baby. It helps us in our conservation of biology. Some species may be more genetically diverse, meaning that they're probably more likely to be protected. And it can show the species interactions between different species that exist. And so genomes are also take measures of evolution and being able to learn about the genomes and the differences between the genomes. We can actually see how related some species are to each other and how long it takes for things to evolve. So molecular biology has added a new measure to the toolkit of evolutionary biology, being able to see how related things are, to see how many different changes there are, and to see how long it took for these species to evolve. And with that, guys, that finishes chapter 5.